Since we are talking about cells, we are going to do a little experiment where we're going to take a look at some DNA. So here are the materials that you'll need.
You'll need water. This could be distilled or spring. You'll also need sodium chloride solution, sodium sulfate solution, ethyl alcohol, a small drinking cup, a 10 milliliter syringe, and a 10 milliliter graduated cylinder. First, you're going to add 1 milliliter of the sodium chloride to the graduated cylinder. Then, you'll measure out 30 milliliters of water into the small drinking cup. Then, swish the water around in your mouth for 30 seconds. Then spit it back out into the drinking cup. Then pour 3 milliliters of the contents into the graduated cylinder. Then add 1 milliliter of sodium sulfate into the graduated cylinder. Gently mix the contents together. Next, hold the test tube at an angle and carefully add 5 milliliters of the ethyl alcohol down the side of the test tube. Then observe. The steps in this lab teach a great deal about the properties of cells, cell membranes, and DNA. When I swished the water around in my mouth, I was collecting cheek cells. These dead cells are being sloughed off continuously. Recent dead cells still contain their nuclei and their DNA. Sometimes this DNA is collected in a forensic situation, and they're analyzed to be traced to a specific individual. In this experiment, we use sodium sulfate. The sodium sulfate breaks down the lipids and proteins that form the cell membrane. So this means that the contents of the cell, including the nucleus, are released from the cell, and then they've become available for further treatment or isolation. The last step requires the ethyl alcohol. DNA is insoluble in alcohol, so when it comes in contact with the alcohol, it dehydrates it and precipitates it.
I hope that you enjoyed my experiment about DNA. All living creatures have DNA, including plants and animals, and it's very interesting to study. So I hope that you enjoyed, and I'll see you guys later. Bye!